So we're out in the field today. This is actually our first day in the field. Last night was our first sampling night. And so we went and collected our samples this morning. We've been processing them all day long. This is part of a National Science Foundation sponsored project, which is related to looking at species that occur at high latitudes or at high elevations. And our focus is on mammals that may be distributed from the tops of mountains in New Mexico and Colorado all the way north to the Arctic Ocean and across Alaska into Siberia. And so we've been working on this question for about 15 years now to try and understand how changing environments, particularly climate change, may be impacting these species at high latitudes and at high elevations. Of course, here in the Southwest, because we have very limited alpine or high elevation species, with climate warming we suspect that many of these species will be under stress. And so we're particularly excited to be able to archive and sample these populations now and over the next several years to see how they may be responding to these new stressors in the environment. So we visit all these locations and we sample the small mammals, and we worked with a number of other scientists. On our field crew today, we've got 3 parasitologists who are specialists in different kinds of parasites, so that every mammal specimen that we collect actually is a host for a variety of other kinds of organisms. And so we're interested not only in looking at the mammal species, but also each of these has all these different parasites and pathogens. And as we slowly begin to study each of these, then each of them tells a different story about their history. My interest is in molecular genetics, and so basically we start some of that work out here in the field by bringing a portable lab. And in the field what we do is, after we collect the sample off of a trap line to sample small mammals, we bring those samples back to the lab, and then we study and dissect various parts of each specimen. So every flea, every tick, every worm that might come from a particular mammalian specimen is tied together so that we can get a very holistic picture of the biology of every individual that's collected. And then all of the tissue types from each of those specimens is put in liquid nitrogen to preserve the molecules so that we can study the genetics of these things later, or we can send samples to virologists so they can study the viruses of these forms. And so basically in our portable laboratory tent we have a setup where every station is somebody who is an expert in preparing a particular preparation that will then go into somebody else's study back at a real laboratory at a university or some other research institute.